Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Biology 2022 Paper 1 Higher Tier for the separate science students. This paper is 1 hour 45 minutes long and it's tested on the 4 topics in biology for paper 1, which is cell biology, organization, infection and response, and bioenergetics. And this paper has 7 questions in total. So let's have a look at question number 1. Question 1 is on cell biology. This question is about cells and transport. Complete table 1. So in table 1, you have to name the part of the cell. The function contains the genetic information. That's the nucleus. You could label that as the chromosome as well. The mitochondria. The function of the mitochondria is where aerobic respiration takes place. So you could call it the site of aerobic respiration. And the part that controls the movement of substances into and out of cells is the cell membrane. Cells in potatoes are plant cells. Cells in potatoes do not contain chloroplast. What is the function of chloroplast? Chloroplast is where photosynthesis happens. You could call it site of photosynthesis, or let's call it site of photosynthesis. Site of photosynthesis. They're mainly looking for the word photosynthesis in your answer. Question 1.3. Name one type of cell in a potato plant that does not contain chloroplast. There's a couple of cells. Any of these would do. The root hair cells, or the xylem and the phloem, or the epidermis, or the meristem. I would just stick to the root hair cell. A student investigated the effect of salt concentration on pieces of potato. So this is actually referring to one of your required practical on osmosis. So this is the method used. Cut 3 pieces of potato of the same size. Record the mass of each potato piece. Add 150 centimeter cube of 0.4 mole per dm cube salt solution to a beaker. Place each potato piece into the beaker. After 30 minutes, remove each potato piece and dry the surface with a paper towel. Record the mass of each potato piece. Repeat steps 1 to 6 using different concentrations of salt solution. What is the independent variable in the investigation? So the independent variable is what you actually change on purpose. So concentration of salt solution is what you are actually changing on purpose. So that would be the independent variable here. So that's my independent variable. The mass of the potato is actually the control variable. If I use mass of potato, actually that would be the dependent variable. Right? Dependent variable. Oops, so that's a bit bigger. And time potato is left in salt solution. That's your control variable. And volume of salt solution is also your control variable. Now, although I say mass of potato piece is your dependent variable, that's the change in mass is actually the dependent variable. 1.5. Why did the student dry the surface of each potato piece with a paper towel in step 5? So in step 5, they dry the potato piece once it's in the salt concentration. When they take it out, they're drying it with a piece of paper towel to remove the excess water. Why is it important? Well, you only want to measure the mass of the water that is actually inside the cells. So to remove excess water and to make sure only the potato mass was measured, or if you could say if the water was left on the potato, then the mass would be higher or it would be affected. 1.6, the student calculated the percentage change in mass of each potato piece. For one potato piece, the starting mass was 2.5 gram and the end mass was 2.7 grams. So the change in mass here is 2.7 minus 2.5. That gives me 0.2 grams. So, or the increase in mass. Calculate the percentage increase in mass of the potato piece. They have given you the equation. So that's 0.2 divided by the starting mass, which is 2.5, and multiply that by 100. That should give you 8%. The student used the results from each potato piece to calculate the mean percentage change in mass at each concentration. Table 2 shows the results. So you have the different concentrations of salt solutions from starting at 0 and increasing by 0.1 mole per dm cube to all the way up to 0.4. The mean percentage change in mass has also been given. Complete figure 1. You should label the x-axis. Use a suitable scale for the x-axis. Plot the data in table 2. Draw a line of best fit for 4 marks. So you could say each of them actually carries 1 mark. So let me have a look at the graph, and let me just bring it into focus so you can see all of that in one screen. There you go. Right. So you have on the y-axis the mean percentage change in mass. So the x-axis should be the concentration. So you can simply copy the label that is actually in the given in the table. You don't have to write your own label. So just literally copied the label that is on the table that was given. So the concentration of salt solution in mole per dm cube. And the next part is actually to put the numbers on the y-axis. So you have, it goes all the way up to 0.4. So let me bring in the table so you can actually see. So that's my concentration. Maybe I can actually bring it over here. There you go. And the mean percentage change in mass. So the labels I would add is there. So let me use a blue color pen. So that's 0.1, 0.2, 0.3, 0.4. You don't need 0.5, but I'm going to go up to 0.5. So at 0, it's 9.8. Now it's 5 boxes for 1, so that's each box is 0.2. So 9.8 should be there. If I make that slightly bigger. Let me see if that works better. Okay, probably a bit slightly more bigger. There you go. Right, 9.8. At 0.1, it's 9.5. So it should be somewhere in the middle over there. Oops, that was a mistake. Let me redo that again. Nine 9.8 should be. So 9.8, 9.5, and then it's 7. 7 is going to be there, and at 0.3 concentration it's 0.4. 0.3 is 0.4, that will be just 2 boxes, and at 0.4 mole per dm cube it's minus 1.4, so it should be just over there. The final part is actually to draw a line of best fit. Now, doing a line of best fit on this is going to be a bit tricky, but let me see if I can do that. So it has to be a smooth line of best fit. It's not going to be a straight line because the points are not, these three may actually look like as if it can actually fall on a straight line, but it looks like it's going to be a curve like that. Right? So let's see if I can draw this. Oops. Oh, that's not going to work. Okay, so here's the line. It's not the perfect, but you should try and get it as smooth as possible. What concentration of salt solution was equal to the concentration of the solution inside the potato piece? So the concentration where it is going to be equal is when there is no mean change. So that's when it's 0, when it's actually crossing the line. So according to the line that I've drawn, it's crossing at the first box. So that's 0.31. Right? Let me just zoom it out for you. Makes it better. So that's 0.31. Yeah. So now, usually the marking scheme will give you a range, and the marking scheme gives me a range of 0.31 to 0.33. So our answer is the line should actually be fine. Next question. Explain why the potato pieces in the 0.4 mole per dm cube salt solution decrease by mass. Now, the only reason that the potato pieces will actually decrease by mass is if it's losing water. Right? Now, if it's losing water, that means that it's actually in a hypertonic solution, meaning that the salt solution is more concentrated outside the cell than compared to the inside. So water actually moves out of the cell by osmosis. So our answer for 3 marks is going to be water moves out of the cell by osmosis. So each of these bullet point carries a mark here. And the last part is the explanation, because the solution in the cells is less concentrated than outside, or because the solution in the cells is more dilute than the outside, or you could say the cell is in a hypertonic solution. Now, it is important that you know what hypertonic, the types of solutions. That's 3 types of solution. So here's a little bit of notes for you. Right? The definitions, right? Isotonic means a solution of equal concentration to the cell. So our answer to this point, this 1.8, right? The isotonic solution is at 0.31 mole per dm cube. That's when there is the net change is actually 0. Hypotonic is a solution of lower concentration than the cell's concentration. And hypertonic is when it's in a higher concentration solution. So what actually happens to cells? Now, you need to remember what actually happens to cells for both animal and in plant cells. So if it was normal human cells, in a hypotonic solution, water moves in. In both plants and animal cells, in animal cells, the cell eventually, it gets bigger and it eventually bursts because there's no cell wall to protect it, so we call it lyased. In plant cells, we call it turgid because the cell wall actually protects the cell from actually burst, so it actually gets firm and actually slightly bigger. In isotonic solution, the cell size is actually maintained in both plant and in animal cells, and the net movement of water is actually the same. In hypertonic solution, in animal cells, the water moves out, which means that the shell actually shrivels up, so we call it shriveled. In plant cells, the cell membrane actually retracts inside, right? The cell actually manages to keep the shape of it because of the cell wall, and we call it plasmolized. That brings us to the end of question number 1, and I will see you in the next video for question number 2.